Hello, my name is Renee Ralston, and I'm presenting our research over the investigation of Bradyrhizobium japonicum as a potential biodegrader for glufosinate. Over the past 50 years, glyphosate has been the most popular herbicide worldwide. However, resistance toward this herbicide has increased exponentially over the past couple of decades. In the US alone, we found 18 strains of glyphosate-resistant weeds, and 20 different countries have already banned or restricted use of glyphosate herbicides. This has caused glyphosate sales to plummet, while inversely caused alternative herbicides sales to increase greatly. The most popular of those being glufosinate, which has gained a lot of traction in the US in the northern and mid-southern areas in soybean fields. Glufosinate targets glutamine synthetase, which is an enzyme that catalyzes the assimilation of ammonia in plants and turns glutamate into glutamine. Glufosinate goes in and competitively inhibits this enzyme, stopping this reaction and causing ammonia to accumulate in the plant cell, eventually leading to plant death due to ammonia toxicity. Now, phosphonitrypsin acetyltransferase, also known as PAT, goes in and acetylates glufosinate and changes its conformation so that it can't go in and bind to that active site, leaving the glutamine synthetase alone, which causes the plants to be able to assimilate ammonia. Our lab has been interested in Bradyrhizobium japonicum simply because it is a soybean symbiont, helping those soybean plants to fix nitrogen. In past mesocosm studies in our lab, we found that Bradyrhizobium japonicum is tolerant to higher concentrations of glufosinate. We then found a whole genome sequence from JGI-IMG of Bradyrhizobium japonicum, which showed us that it has the PAT gene in it. We then did a nucleotide blast of the PAT gene and found that it had 100% similarity to Bradyrhizobium japonicum. After that, we performed a protein blast of the PAT gene and found that it had a lot of similarities to other PAT genes in different organisms that had already established their tolerance to glufosinate. In our most recent study, we grew a starter culture of Bradyrhizobium japonicum, made a glufosinate solution, and prepared two different medias, one of which had a simple source of nitrogen and a complex source of nitrogen, and the other of which had just a complex source of nitrogen. So we did a random map of the 96 well plate and then inoculated that 96 well plate with controls and 3 different types of media, which were all 3 were inoculated with B. japonica. The first had both the simple source of nitrogen and the complex source of nitrogen. The second had just the complex source of nitrogen, and the third had a complex source of nitrogen and 200 micromolar of glufosinate. After performing the growth curve assay and having it take recordings every hour and a half for over 55 hours, we found that the media with the complex source of nitrogen and glufosinate had the best growth out of the 3 medias that we tested. This supported our hypothesis that glufosinate could potentially degrade, that Bradyrhizobium japonicum could potentially degrade glufosinate and use it as an alternative nitrogen source. For future work, we plan to do longer growth studies and more enzymatic assays for the functional conformation of the PAT gene in Bradyrhizobium japonicum. Thank you so much for listening, and I hope you all have a great one.